it up for Josh.
So the paper I'm presenting on today is targeted DNA degradation using a CRISPR device stably carried in the host genome. Or in simpler words, destruction of DNA using a CRISPR device. So I'm going to start out with an open-ended question to you guys, which is, how have researchers destroyed DNA in the past, and secondly, why do you think it matters?
Josh, why would you need to destroy DNA?
Okay, so I'm just going to answer this question. So, when we took the cancer class, obviously, researchers would want to destroy DNA, and then thus would induce apoptosis or cell death, and thereby reducing the tumor size. So it has an application in that field. But another aspect that I want to focus on is GMOs, or genetically modified organisms. So to create synthetic DNA, it takes a lot of time and it takes a lot of money. And the effort and resources that it truly takes to create the synthetic DNA and finding a specific gene that creates an advantageous feature in a new organism is incredible. So when you introduce the gene into the new organism, you would want that property almost to be yours, like a patent. So it would be very unfortunate if other people would be able to get their hands on it, other people would be able to access that synthetic DNA when you had spent all that time and all that money creating it and introducing it into the new organism. So, and also I'm just going to answer the question, how researchers destroyed DNA in the past. So in the past, researchers had used restriction enzymes and nucleases, but the problem with those methods was that it was non-specific, so you would have a lot of off-target interaction on the genome, and you would end up cutting off and interacting with other parts of the gene that weren't necessarily targeted, and that would harm the overall genome and the status of the cell. So now I'm going to explain what is CRISPR. So CRISPR was a mechanism adopted by bacteria to prevent against viruses and viral DNA being inserted into their genomes. And it's basically that a guide RNA will lead a Cas9 nuclease to the target sequence. A guide RNA is basically a guide molecule for the Cas9 nuclease. Once again, the nuclease is just a molecule that will cleave the DNA at a specific location on the genome. Once the guide RNA leads it to that viral DNA, it will lock on to what is called the PAM sequence, which is just three base pairs that will serve as the recognition base pairs. So that is going to help the guide RNA and the Cas9 nuclease identify where to separate the two DNA strands and where to cleave it. And I put an asterisk next to the PAM because I'm going to come back to that later. Once it does that, it will cleave the DNA at that location. Usually it's to replace that target sequence with a new sequence, and that's where we get into genetic modification. But in what I'm about to talk about and in what the researchers studied, it is purely just to degrade the DNA. So now what is DNAI? DNAI is CRISPR-mediated degradation. It adopts a lot of the same mechanisms and a lot of the same practices of CRISPR to enact very similar results. Most importantly, it's a stable device. And as I said, in the past, researchers have used the non-specific nucleases that were not necessarily very specific at cleaving the DNA at a specific target, and it would have a lot of off-target interaction. When it was in the off stage, it would have further interaction when it necessarily wanted to. DNAI is based on a transcriptional input, so you're able to give the device any target sequence to degrade, and it results in rapid cell death. So when you give the DNAI device a target sequence to degrade, it will not necessarily harm the overall status of the cell, but if you give the DNAI, when you give DNAI the order to degrade the entire plasmid, it will result in rapid cell death. And I'm going to get into that later a about how that can have applications within environmentalism. So now can anyone tell me how DNAI and some of its aspects are similar to CRISPR in any way? Might have to call somebody out. Jackson?
I mean, it seems like DNAI doesn't use CRISPR. Or you said use CRISPR-mediated degradation.
Yeah. So the CRISPR mechanism is already native within the bacteria cells, so they're just adopting the same exact mechanism, the same exact technology, and they're just using it for a different application. It's the same technology, but now they're just using it to destroy a specific target sequence or destroy the plasmid. So degradation of the target. The effect of the DNAI device is dependent upon two different factors in this experiment. One is the spacers, which will be the targets, and two is the actuators, which will be like the motors that will drive the DNAI device to the genome. So a lot of stuff is going on at the X and Y axes, so I'm just going to explain what's going on there. So here first you have DNAI device, whether it's off or on, and you can see pretty consistently throughout, black is control and white is when the DNAI device was turned on and they were looking for results. And you can see consistently throughout 10 to the 0 power, which means 1, and here you see fraction target cells. So you can see that when the DNAI device is turned off, it has no effect on the overall status of the cell, and it will not degrade the genome or the target in any way. So that further accentuates that this is a very stable device, and it's not going to have off-target interaction, and it's not going to have interaction when the researchers don't want it to. Here you have spacers, which once again are the targets, and you have spacers N, Y, Z, and Y plus Z, so they just combine the Y and Z spacers. The N spacer is the control, and the Y and Z spacers is what they experimented with. And then the actuators. So you have 1 times for these 4 sections, and then you have 3 times here. So you have 1 actuator in these 4 sections, and you have 3 times the actuator in these sections. So what I really want you to focus on is what's in this red box here. And what the researchers were basically focused on, how can we make this device as effective as possible? How can we combine certain characteristics and certain molecules in certain ways that will make this device the most effective in degrading the DNA? So they determined that combining the Y and Z spacers and producing 3 times the amount of actuators would do that. As you can see, 10 to the minus 8, that's a very, very small size. It degraded the DNA into a small fraction of what it originally was, as opposed to these other experiments. And what I want to focus on is dose dependency. So if you have increased factor concentration, you're going to have increased effectiveness of the device. So before they were looking at degradation of a certain target, a certain location on the plasmid, now they focused on a broader perspective. Can we destroy the entire plasmid? So what I want you to get out of this graph is, first off, that they can destroy the entire plasmid, and second off, a time chart. So after the 2.25 hour mark where the device was activated, you can see within 6 hours how effective it is to, once again, reducing the plasmid size to 10 to the minus 8. So once again, it reduces it to a very, very small size. So right here, I think it would be fitting to compare this device almost to a paper shredder. So now they focused on, okay, we can destroy the DNA and we can eliminate it to a very, very small size to what originally it was. But what if people are able to re-amplify that DNA and regrow it to the status it was before they were able to degrade it? So right now I want to focus on the pVac target. So here, off and on, with the control N and with the spacers Y and Z, and on the y-axis, absolute copy number. So that's how many copies of DNA were they able to regrow from what they were able to degrade in the last experiment with the plasmids. So you can see, with another method, they were able to show that the absolute copy number was able to be regrown to around 10 to the 9th power. But when they used the DNAI device, it was able to be regrown to only 10 to the 5th power. So, once again, with the paper shredder analogy, the paper shredder, it's basically like if you take DNA as paper and then you use the DNAI device, it's almost like a paper shredder. It's going to be almost impossible to get all those shreds and put the DNA back together. But if you use another method, it's almost like you're crumbling up the piece of paper and throwing it away. Even though that might help in destroying the DNA and getting rid of it, it's not going to be as effective in just totally shredding it. You're never going to really get the DNA back to what it was before. So once again, we told you to look out for the PAM sequence. So the strength of the PAM is going to influence the DNA degradation. Once again, PAM is 3 base pairs that will help the DNAI device and the guide RNA determine where to lock onto the genome, and it will serve as like the recognition base pairs for where to cleave the DNA. So on the y-axis you have the fraction of the target cells, and on the x-axis you have the time post-activation. Once again, the device is activated at the 2.25 hour mark. With the black lines and the blue lines, you have strong PAMs, and you have AAG, AGG, ATG, and GAG. With the teal turquoise lines, you have the weak PAMs, and then with the CCG, that's basically your control. So you can see that having a stronger PAM sequence is going to influence how effective the DNA degradation is.
Did they talk about what makes a strong PAM or weak PAM?
Yeah, so what makes a strong PAM is going to be how attracted the DNAI device is to the genome. So basically pluses and minuses, how greatly they interact and how easy it is for that device to cling on to the genome and to be attracted to it. So I just want to make a distinction here, because this can get a little confusing, is that the inducer is the activator and the inducer is not the actuator. So while the actuator is the motor that will drive the DNAI device to the cell, the inducer is going to be the activator of that actuator. So just another analogy for like a car analogy, the actuator is going to be like the motor of the car, and the inducer can be like the oil that serves as the inducer, and it's going to give it that activation. So what the researchers used as their inducer is arabinose, and you can see here 0, 2, and 10 millimoles of arabinose. And then when the DNAI device was turned off, and then when the DNAI device was turned on. And here on the y-axis, you see viable cell titer, and they measured DNA degradation as a function of the cell density, or the cell titer. So here you can see the black bars where it's control, and the white bars where they experimented with the device. And you can see that even though how much you increase the inducer, it's pretty much going to hover around 1.0 for the viable cell titer, symbolizing that DNA degradation is pretty constant no matter how much you increase the amount of the inducer. But when you increase it over here, you can see that there's a steady decrease in the viable cell titer, symbolizing that there is greater DNA degradation, and that is a function of the inducer. And once again, in the overall scheme of why would they do this, we're trying to figure out how can we make the device as effective as possible. And once again, it was dose dependent. So the more amount of inducer you had, the greater amount of degradation you had. So DNAI is a CRISPR-mediated device, which employs a lot of the same technology as CRISPR. It uses the PAM sequences, it uses the guide RNA, and CRISPR is used in a lot of different ways, and the researchers just wanted to see how can we use CRISPR in another way that we haven't thought about before. It destroys targeted DNA, and it does so with very high specificity. So, as I said before, in the past when researchers had used the non-specific nucleases, it had cleaved the DNA, but it had a lot of off-target interactions that had not necessarily been effective at completely degrading the DNA and doing so at a specific location, and that would affect the overall genome when the researchers had not wanted that to happen. And it is dependent upon different factors. So the spacers, the actuators, the inducers, all of these are going to affect the overall effectiveness effectiveness of the DNA degradation, and once again, it's dose dependent. So for the most part, the more you increase the amount of factor, the more effective the DNA degradation is going to be. So now I'm going to focus on how could this device, something in biotechnology that you probably have never heard about before, how could this affect your lives? So it is going to protect intellectual property. As I talked about before, when you create synthetic DNA with GMOs, it's basically like filing a patent. You would want that specific gene, that specific synthetic DNA to be yours, and you would want all the benefits of having that, all the positive characteristics it might create for a certain organism to be yours, and if that got into the hands of the other people, you would want that DNA to be able to degrade itself so other people wouldn't be able to access it. As I talked about before, this can help with environmental problems and it poses a solution to environmental concerns. So if you have a release of bacteria in a certain industrial or lab setting, there are certain ways that you can get rid of that bacteria, get rid of that release. One of the ways is pesticides, but that's also a way that can harm the environment. So having the DNA, the organism being able to degrade itself and thereby inducing apoptosis within those bacteria cells, it can just immediately induce cell death, and all the bacteria that was caused in the release would be able to be circumvented. Yeah.
It's basically like a self-destruct button
Yeah.
that you can put in and out.
So it's going to be based on cell signals, and based on the transcriptional input that I talked about before, you can program this device to recognize, okay, I was released from a specific medium, I was released from a specific environment, okay, activate the DNAI device, and then it's going to cause the DNA degradation and then cell death, which is really cool. It's also a new way to deactivate genes. So we had talked about before with stuff like RNAi and epigenetics, but this is a new way that researchers hadn't really talked about before, using CRISPR to deactivate the genes. So instead of worrying about whether those genes would be reactivated somehow, now there's no more gene to worry about because it was just destroyed. And finally, this is going to advance the field of biopharmaceuticals and CRISPR. So a few weeks ago, we had Jake talk about how CRISPR can be used to control the spread of Zika virus. And CRISPR, as we probably know, is a very powerful tool, and it has, even though it can get out of hand, it is something that we should really utilize and something we should really cherish because it is one of the guiding technologies of science today. And advancing the field of biopharmaceuticals. So even though these researchers had focused on actuators and spacers and inducers, maybe there's something that they hadn't really focused on that can affect the overall effectiveness of this device. And further experimentation, hopefully, in the biopharmaceuticals and biotechnology industries will yield results on how to make this more effective. And that's it. And just a big thank you to the journal